Okay, so now we will do the warm-up. So being stuck at home has got you feeling down, so you decided to start a garden. You till a small plot of land in your backyard and plant 4 types of seeds: rose, tulip, daisy, and sunflower. The flowers bloom a few weeks later. So we're going to go through the warm-up tab on your Excel workbook for Lab 2 and complete the problems with the data there.
Okay, so for the first one, we've got from columns 2 through 4, row 2, we're keeping row 2 constant, and we're square rooting the value before we sum them. So we've got the sum of the square root of row 2, columns 2 through 4. Close parenthesis, enter. Another way to do this, instead of having these 2 functions embedded in each other, right? So we're square rooting first and then summing. You read from inside to outside, is to make a square root table. Should be, so equals the square root of this one. Because remember, I think I mentioned before that Excel works in a spatial orientation kind of way. So it memorizes essentially the relationship between this cell, L2, and the one that I'm referencing, A2, or sorry, L1 and A2, yeah. So I now find the square root of cell A2. And if I click and drag, which is what we were working on last week, I can make a square root table that is the square root of each of these corresponding values. And then I can do equals the sum of row 2, columns 3 through 4. For number 2, we've got rows 3 through 5, columns 3 through 4. No functions being done. So the sum, remember the equal sign in front of it, the sum of rows 3 through 5, so these 3 right here, but only columns 3 and 4. So this data set, or chunk of the data set right there. Close parenthesis, enter. For number 3, this one's a little bit more complicated, and there are a couple of ways to do this. I don't think that your version has this sum squared function, but I'll go over that in 1 second. So one way to go about this, if we're looking at rows 1 through 5, columns 1 through 4, that's the entire data set. But before we sum everything, we want to add 2 and then square it. So one way to do that would be to do equals the sum of the whole data set. Oh, actually, I missed a parenthesis. Open a parenthesis again of the whole data set plus 2, close parenthesis, caret 2, close parenthesis. And this reads as from A2 to D6, so all of these, individually add 2 to each of them, and then square the entire thing, and then sum them together. Another option would be to do the same thing we did up here, to make a table first. And I'll actually make it a little bit easier to read. I'll put this row up top. And this is going to be a +2 and squared table. So equals, open parenthesis, A2 plus 2, close parenthesis, squared. So we're adding 2 to the variate and then squaring that value. I can now drag it over and then drag it down. And I now have a table that has each variate +2 and squared. So now using that new table, I can sum, so equals sum, open parenthesis, the whole set, close parenthesis, enter, and I get the same number.
Kyra,
Yes.
what is it the instant where you're like it's what am I trying to say? You're adding it by 2, like the entire data table, and then you're squaring the entire data table. Like, when is it separate with the parentheses? Like, when you're supposed to do it after you do all the summation?
Like, how would that look, or like when's the time you would do that?
Yeah, so when I first had looked at this, I thought you were just adding all the value, like all the values were added by 2, and then you squared whatever that sum was.
Okay. So if that was what this was, there would be, I hope you can see this. There would be parentheses like here, and the square would be up here.
Okay, that makes much more sense. Okay.
Yeah. Okay. And then up here in the function line, you should be able to see the equation for that first one. And then there is one other way that you can do this that another student pointed out using the sum squared function. So we use equals sum squared. I think it was actually a student in this class last week. So thank you, whoever that was. Sum squared. So this takes the sum of squared values. So we can do the sum. You can select the entire data set +2, close parenthesis, enter. And we skip that additional step of having to do the caret 2 like we did in the first example, or the first method. So those are 3 different ways of getting the same answers. Are there any questions, or does anybody want me to hover over any of those functions first? Okay. So number 4, what's the equation for the sum of all roses and all tulips? So roses and tulips, that would be rows 1 through 5 and columns 1 and 2. So that would be the sum of roses and tulips. Close parenthesis, enter. And the equation for that is, so we've got the sum, oh, hold on. We've got the sum for rows 1 through 5 or columns 1 and 2. It doesn't really matter which summation you put first. I'll do columns first. So columns 1 and 2 and the sum for rows 1 through 5 of all variance. And then number 5, what type of data are these? Are they discrete, continuous, nominal, etc.? These are discrete, right? They are just whole integers. If we had like 6.5, then we would have continuous. We would be considering the infinite numbers between 5 or 6 and 7, right? But because they are whole count numbers, these are discrete. They're also nominal because we have 4 nominal or categorical or attribute categories. Are there any questions about the warm-up? Okay. It looks like some people might be having error messages coming up because of the summations. My class on Monday also had this issue, and the student was able to solve it by uninstalling Excel, reinstalling Excel, and then updating Excel. So I'm going to recommend that as the option. I spent I don't know, a lot of time this past week trying to figure out what was going wrong, and it seems like that might be the issue. So if you're having that and you're getting like this like hash value error message, I recommend you try doing that after class.
And I actually have a question. I'm sorry.
No, it's okay. Go ahead.
So for the warm up, so for number 4, when you wrote, like X, I, J, does it matter if you put a comma between the I and the J or no?
No, it doesn't.
Okay, sweet, thank you.
Yeah, it's kind of just a personal preference whether you want it there or not. I think it starts getting a little clunky because you're already writing so small.
Yeah.
That having the comma in there can be a lot, but yeah, it's up to you whether you want it there.
Thank you.
Yep, you're welcome. Okay. So if there aren't any other questions, we'll go ahead and start discussing data displays. So, okay. So data displays or visualizations or figures or whatever you want to call them are really, really important for showing the data visually, right? As humans, we're really not very good at understanding patterns in data when we just look at numbers, right? If some patterns are kind of obvious, it might be easier, but generally we're not good at it. So we use data visualizations and displays to do that so that we know what kind of analyses we want to use. It's often the first step in the data analysis process. So a little review of data, types of data or variables. We have categorical and numerical. Those are kind of the two really broad categories. Categorical is the same thing as attribute or nominal, and it's just a characteristic variable being recorded. So color, sex, age class. A numerical value is just a number, right? And these are the continuous, discrete, or ranked. So there's different types of numerical values, but the important part is that it is a number. When we're looking at categorical data, we have count data and we have means or continuous data, right? For the count data, we have discrete variables and we only have one number belonging to each category. So in this example here, we have females and males, and then we have visual acuity, right? We have colorblind, a carrier for colorblindness, or not colorblind at all. These are whole numbers out of the number of people sampled. and they're just people, right? There's only a single variable being considered here. Same for over here, right? We have, in this example, we're looking at light regime and days since picking. I can't remember exactly what this experiment is supposed to be, but we have 2 light regimes and 3 days since picking, and we have several numbers actually in each category. And this immediately tells me that we're going to be analyzing means. We're going to be taking the average of each of these groups and comparing them to each other. Same for over here. We've got 3 groups with continuous variables, continuous numbers in between. This data automatically, I'm like, we're comparing the means between oyster reef, bare ground, and a concrete block to see how much of whatever this is has grown. And an important thing to note here is that all of the numbers visible are the same variable. We don't have multiple variables in the data set. We have, you know, different explanatory variables, male, female, color blindness, so on and so forth, but the actual data values are all of the same type of measurement. And these will look familiar in the future. Right now, we've kind of set this up to show you different examples of data that you're going to see in this class. Not important to you now, but this would be a chi-square, this would be a 2-way ANOVA, and this would be a 1-way ANOVA. There's very specific data setups that kind of cue to you which type of analysis it's set up for. We're looking at numerical data, so we're not looking at is a category causing a difference between something, right? Because that's what we're looking at here. We're saying male versus female, low versus high, days 1 versus day 3, right? Here we're not looking at speed versus pulse. We're looking at how do these change together? How does one changing maybe relate to the changing of another? The big question when we're looking at data like this is, is one of the variables being controlled? If one of the variables is being controlled, then we actually have the power to say that something is being caused, right? So here we control for the speed of the bike to see how the pulse responds. And this gives us the power to say that speed causes pulse to change because I'm controlling this to see how this responds. If neither of the variables are being controlled, then we don't have that power. We can only say that 2 variables maybe change together. They're correlated to each other. So here we're not controlling the weight of a sand crab or the number of eggs they carry. We're just seeing, does weight happen to change the way that eggs, the number of eggs carried? And different from the previous slide, the numbers here are of different variables, right? Speed is in 1 unit, pulse is in another unit, weight is in 1 unit, eggs are in another unit. And again, looking at these, this would cue me that I'm going to do a regression or a correlation. You'll learn that later on though. When we're making displays for frequency distributions, if we have categorical data, one way we might do that is a bar chart or a frequency table. If we have numerical data, we might use a histogram. I've got a couple of slides here that are all pretty redundant, but I've included them all because, you know, different things help different people learn. Different visualizations help for different things. When we're displaying the associations between variables, we've got a couple of different options, depending on whether the explanatory variable or the response variable is categorical or numerical. Explanatory variable is just another way of saying the independent variable or the y variable. And response variable is just another way of saying the dependent variable or the y variable. So if our explanatory variable is a category, is categorical, and the response is categorical, we might make a grouped bar chart or a mosaic plot. And I have examples of this on the next slide. If the independent variable, explanatory variable, X variable is categorical and the response is numerical, we might want to make a box and whisker or a bar chart. If the explanatory variable is numerical and the response is numerical, we're going to make a scatter plot. And then less common, if the explanatory variable is numerical and the response is categorical, we have to do this kind of weird thing where we bin numerical values and show average response using stack bar chart or scatter plots. This isn't super common. I'm not going to ask you to do this, but I included it in this chart just for completeness. Here's another way of looking at this. So here we have, do we have categorical data? First question, yes or no? If you do, is the data count data, or are you comparing the means of groups? If it's count data, a histogram or a stacked bar chart, these two examples right here, are probably what you want to use. If you're comparing means, you should probably use a bar chart or a box plot. I've got some asterisks next to this box plot because there's some pretty good arguments against using a box plot for this. Primarily that box plots show medians, they show quartiles, right? So this thick line in the center is the median or the second quartile. This line up here is the third quartile, this is the first quartile. This is the minimum, maximum, these dots are the outliers. So that's not really super great if we're trying to compare means, right? Means and medians are oftentimes in a normal distribution very similar to each other, but they're not the same thing. So that's the argument for using bar charts and against using box plots. Bar charts will have the categories, the mean represented by the bar, and then a standard error or standard deviation error bars at the top. And these are super important. These show how much variation there is between the data points. So make sure, you got to make sure to include error bars on these figures. Okay, so going back to the beginning, what if we don't have categorical data? Well, is one of the variables being controlled? No. If not, then we're going to use a scatter plot without a regression line, because we don't have the power to say that X is causing Y. That's pretty much what the line implies. So if we are controlling it, we can use a regression line. And this line implies that at any variable of X, at any number of X, I can predict Y because X causes Y. And then a quick note on the difference between histograms and bar charts. So a histogram shows a frequency distribution where the explanatory variable is binned and the response variable is shown as a frequency or a count, an actual whole number count of occurrences in each bin. Bar charts show comparisons between variables and between groups, and the bars typically represent the means, not counts. And like I said before, they have standard error bars. So data visualizations for count data. So again, we've got a histogram and a stacked bar chart. And these are the corresponding data. So here we've got male and female, and then level of color blindness, right? Here we've got colorblind, carrier, normal vision. And looking at this on the x-axis, we can see that this is the proportion or percent of people that have this. So we see not many women compared to men or females compared to males are colorblind, but females are the sole carrier of the color blindness gene. And then females and males are pretty equally distributed with normal vision, maybe a little bit less for males. These are nice to see the proportions split up this way. It's very easy to tell how things split up. One thing that makes it a little bit more difficult though is it's kind of hard to tell, right? For this one, I'm like, oh, it's probably like 22, 23%, but it's a little bit harder for the male to tell what percentage that is, especially when you've got multiple colors and stacks and things going on. Here we've got a histogram where we've got age groups, right? So we have binned data. We have 0 to 9, 10 to 19, so on and so forth, and the number of people that fit into each of those groups. And we have that represented here. With frequency, our counts are whole number counts of how many people fit into each of those groups on the x-axis, or sorry, on the y-axis. Sorry, I probably misspoke over here too. We're looking at data visualization for means and medians. I included this medians because of the box plot, but I didn't show any examples of box plots down here. So we've got the data from before. So we've got light regime, low versus high, Dayson's picking. So here is a visualization for the above data. We're looking at the means of each of these groups and their standard errors. So we've got day 1, low versus high, day 2, low versus high. And this is really useful for looking at, is there a relationship? Is there a interesting interaction between Dayson's picking and high versus low? Here we could say actually maybe, right? At day 1, we see that there is higher average sugar content at day 1 if there was a low light regime. But after that, compared to the high light regime, after that we see that pattern switches. So there might be an interesting interaction going on here. Over here, we've got the oyster reef versus bare ground versus concrete block. Looks like we're looking at Shannon's diversity index by habitat. So we can see that the oyster reef has a greater diversity compared to bare ground and concrete block, but that the concrete block has higher diversity compared to bare ground. These figures always have error bars, and I'll show you how to calculate them later. When we have numerical data, 2 numerical categories, or not categories, 2 numerical columns, and we're seeing the relationship between the two. So we have speed and pulse, weight and eggs. Here, remember we were controlling the speed of the bike so that we can say that increased speed causes increased pulse. That's why we have this regression line on here. Here, we didn't control weight or eggs, so we can't put a regression line on, and we can only say maybe there's a correlation between them. Looks like maybe a weak positive correlation between these two. Sorry, my dog was trying to leave the room. Okay, so these examples are, the next 2 examples are kind of weird, and I'm not really gonna have you do these, but just thought I would show you them. So here we've got a continuous explanatory variable and a categorical response. So we've got a different dose of some chemical or some agent from 0.03 to 0.87. And we're asking, did whatever this is being tested on, probably flies, did they survive or not? So we've got yes or no. We can't really do a whole lot of analysis on this yes or no, so we change that into a code where 1 is yes and 0 is no. We then summarize this raw data into a table like this, where we create bins. So now we have a bin from 0 to 0.9, that would be these 2 values. We have 0.2 to 0.29, that would be these 3 indexes. indices. We've got 0.4 to 0.49. We these these three indices, so on and so forth. And then we are actually averaging these groups. So from 0.03 to 0.04, the average between 1 and 1 is 1, right? So on and so forth until we get to the bottom. And then we also calculate this midpoint. So what is the midpoint between 0 and 0.9? 0.2, 0.29, so on and so forth. And then we actually graph these two columns. The midpoint versus average survival for that binned group. And we can get a scatter plot like this. The reason that we do this instead of just plotting this raw data, right? So ignoring this column, but if we plotted dose and code, what we would get is we get a lot of points, but we'd get them all up here or all down here. And it'd be really hard to see the trend because it would just be up and down all the way across. By calculating the averages and using bins like this, we can actually much more clearly see this downward trend in survival as dose increases. Here's another example of where our explanatory variable is numerical and the response is categorical. So again, we have to bin the data. But this time we're showing the data using a stacked bar graph that shows the proportion or percentage of responses at each income midpoint. So we've got agree, neutral, or disagree for this survey or questionnaire, and the midpoint of these income brackets. And we can see, I mean, I don't know what question this was, but it looks like a pretty interesting trend where people who make a lot more money agree with whatever this question is compared to those who do not. Agree a lot more with whatever this question is. Whereas people at this lower income point disagree pretty strongly with whatever this question is. So this is another example of how you could visualize this data. Okay, so now we're going to switch and I'm going to do some practice examples in Excel. But first I'm going to go over this Budden et al. 2016 paper and pretty much just summarize what it meant, because there was a lot of information in it. But first, actually, are there any questions about that? I know that was a lot of information and I think it'll make a little bit more sense in a bit. I also put a lot of notes in the notes section of the PowerPoint so that you can look back at that and read over anything if you were confused. Okay, if there are no questions, then I'm going to go ahead and just share the paper and kind of walk you through it. So the paper is on the viscous control of cellular respiration by membrane lipid composition. So there are a lot of figures in this paper, right? We've got a ton of figures. So let's kind of walk through a couple of these. And first off, let me just summarize actually what was found. So pretty much the fluidity of a membrane is dependent on the ratio of saturated to unsaturated lipids. More unsaturated lipids equals more fluid, right? A saturated liquid or a saturated lipid would be like butter, right? So it's thick, it doesn't flow, it doesn't run. whereas an unsaturated fatty acid or lipid would be like olive oil, which flows pretty easily at room temperature. So more unsaturated lipids equals a more fluid membrane. The electron transport chain, as a lot of you should remember from general bio, is embedded in the lipid membrane. Therefore, if the membrane is more fluid, molecules can move faster, which means that more ATP is produced and oxygen consumption increases. So if we look at figures, okay, yeah, 2C and 2E are these ones I want to point out first. So these show that as unsaturated fatty acids or lipids increase, so does respiration. Right? There's this positive relationship between the percent of unsaturated fatty acids or lipids and respiration rate. We can see that in both of these. This FABH2 is just a gene that pretty much makes lipids less saturated, more unsaturated. So the first example that we're going to go over in data displays is based on this principle. Second, we find that respiration rates across different cell membranes, so the membranes found in isolated mitochondria, prokaryotes, and eukaryotes, are different, right? So that's another thing this paper was looking at. For the isolated mitochondria, they are very similar to prokaryotes, which was what we would expect, right? So mitochondria used to be kind of just prokaryotes until they were engulfed by a eukaryote, and now they exist in eukaryotes, right? It was the whole theory of endosymbiosis. So we expect mitochondria and prokaryotes to be very similar in this respect. When we look at figures 2E, 3D, where is 3D again? Oh yeah, 3D and 4B, right? Yes. We find that eukaryotes have greater oxygen consumption compared to isolated mitochondria and prokaryotes. So that's what our data display 2 will be based off of. And then finally, the paper also mentions that lipids, like cholesterols, can impact lipid membrane fluidity. Prokaryotes don't incorporate cholesterols into their membranes. They use something called hapenoids instead. It's similar, but they're different. Similar membrane structures are found between prokaryotes and mitochondria. Again, that makes sense because they have more closely related evolutionary histories. The example display that I'm going to show you in a second, the third one, is based on that principle. There's no figures for that. We just kind of made up data based on that principle. And yeah, so this is a pretty scary looking paper, but it's actually not that complicated once you break it down. So go ahead and open up to the data display example 1 in the Excel workbook. You should be able to, I believe, see both my share and your Excel. So go ahead and do that, and we can walk through this together. So for data display example 1, we've got the UFA concentration, the unsaturated fatty acid concentration, and O2 consumption, right? And like I was saying before, we expect to find that as unsaturated fatty acids go up in concentration, as membranes become more fluid, more liquid, that oxygen consumption should increase because the electron transport chain is moving faster and ATP is being produced at a faster rate. Therefore, oxygen consumption goes up. So to make a figure for this, we just go up to insert. First, we want to select the data that we're interested in. And then, select the data, go up to insert. You could select recommended and it'll give you a whole bunch of stuff. I know looking at this though, at the type of data that we have two continuous variables or two numeric variables of different types. So unsaturated fatty acids are 1 unit, oxygen consumption in a 2nd unit. That means we're comparing these two numerical values. We're going to make a scatter plot. So here we have our scatter plot. We're not going to include a regression line because we're not controlling either of these. We're just observing a correlation or a relationship between the variables. This title is not super helpful, so I'm going to change it. I'm going to say oxygen consumption versus, or actually, yeah, let's do UFA concentration versus O2 consumption. It's not the best title, but it'll work. Or consumption, sorry. Consumption. Okay. This is also not a super great figure. We need some axes labels. So I'm gonna click this, add chart element, add axes titles, and rename these. So over here we have O2 consumption, and down here we have UFA concentration. Yeah, and looking at that, I'd say we have a pretty complete looking figure. It's not the prettiest, it could definitely look nicer, but it's not bad. Looking at our second example, we've got oxygen consumption, and we've got our 3 different types of cells, right? We have eukaryote, prokaryote, and isolated mitochondria. We expect eukaryotes to have greater oxygen consumption compared to prokaryotes and isolated mitochondria. Looking at this data, we have the same measurements between 3 categories. I think we're going to compare means, right? I also kind of gave you a little hint because this is down here. So first, I need to calculate the means for each of these groups. We're going to do that using equals average. And then click and select the data, close parenthesis, enter. Now I could do that again here. Well, actually, let's do these first. So we've calculated the mean of the eukaryote group. Next, we're going to calculate something called the standard deviation. So the standard deviation, we're going to get into that not next week, but the week after that, I believe. And standard deviation is pretty much just a measurement, like a single number that kind of sums up how much variation there is in data. Let me draw a quick figure actually. So to kind of visualize this, kind of in following suit with the theme of this class, let's say we've got a normal distribution. The center is the sample mean. So, right, the mean is the most common value, which we've got here. And then we've got less common values at the two tail ends. What standard deviation pretty much tells us is what's the variation in the data. So in this example, I'll draw it over here. We've got the mean in the center. We have a small standard deviation because there's not a lot of variation in the data. Compared to this one over here, we have a much higher standard deviation. So that's pretty much all that standard deviation tells us, is just how much spread is there in the data. And we can calculate that using equals dev.s. There's two of them. There's .p and .s. .p stands for population, and you use it when you're sampling a whole population. And what that really means is that you've taken measurements from every single individual possible, which practically never happens. And so we're not going to use it at all. Realistically, you are always sampling a sample. You're only just taking a couple individuals, right? So in this example, the researchers didn't sample every single eukaryote in the body or whatever. They just took a couple. So we're going to use this .s, this sample standard deviation. But we use it the same way. We just select all of the data, close parenthesis, enter. Next, we're going to calculate something called the standard error. And the standard error is pretty much the same thing as standard deviation, but it's corrected for the sample size. And it's equal to the standard deviation divided by the square root of the sample size. And I've got a little bit more information about this over on this new functions tab. So these are all the functions we're going to use in class today: the average, standard deviation, square root, count. And I've also got the equation for standard error here, written here and explained a little bit more. But yeah, so it's just the standard deviation divided by the square root of the sample size. So calculating that here, we're going to do equals, click the cell for the standard deviation. We don't have to type it out again. Divided by the square root, SQRT, open parenthesis, and then the number, the sample size, right? So I could just count all of these, like 1, 2, 3, 4, 5, 6, so on and so forth, right, to get what the sample size is. Another really useful function, though, is count, which counts the number of cells you have selected. So I'm going to use count and then select all of the numbers, and that will count how many cells there are. Close parenthesis, enter. And we get that the standard error is 27. So I'm gonna go over standard error one more time, because I think in my other class a lot of students were really confused, understandably. So standard error is standard deviation adjusted for sample size. And the example, or kind of like thought experiment for that, is if we have a standard deviation of 96, but our sample size is like 4,000, that's not a lot of variation, right? For sampling 4,000 individuals, you would expect there to be a lot of variation. So let's do that really quick, right? If we had a sample size of 4,000, we would do the standard deviation divided by the square root, oh, I don't have to do count, of just 4,000, a huge sample size, right? Now our standard error is very small. It's like saying like wow, you took a lot of samples. Standard deviation of 96 is actually not that much considering how much variation there could have been. Our actual sample size in this example is pretty small, so we didn't get to divide our standard deviation by much, and so we still have a pretty large standard error. Okay. And like I said, we're going to talk a lot more about standard deviation, standard error, all that stuff in a couple of weeks. But continuing on, I could type all of those things again, but instead I'm just going to select all of these and drag to the side to calculate everything. These values, mean and standard error, are what I'm actually going to use to make the figure, not this stuff up here. So I'm going to highlight this part first. Insert, I want a bar chart. And there we go. This is missing standard error bars though. So I'm going to click this plus to add chart elements. I think if you're on a Mac, you can just find it up here, this add chart element up here, and do error bars from there. It's another option. Regardless, so if you do it that way, you're going to select more error bar options. If you do it this way, you're going to select error bars, click this arrow, and then more options. When we get over to this more options area, we want both. We want upper and lower standard error. Well, we want to select custom, not standard error. I know it seems like you should click standard error. Don't do it. We're going to click custom and then specify value. And you'll get this pop-up window. Gonna delete the positive error value. Positive and negative just means above the bar and below the bar. Delete that. Select our standard error that we calculated. Do the same thing for the negative. Delete that. Select our standard error. Click OK. And there we go. Now we have correct standard error bars. Again, I'm going to change the title. So mean O2 consumption by cell type.
I'm sorry.
I'm gonna-
I'm sorry. Can you do that again about the error values that you used for the
Yes.
please? Sorry.
No, it's okay. Okay, so let me just delete them and I'll just like do it again from the beginning. So this will be pretty much the figure that you'll get initially. That I changed the title, obviously. So you click on your figure, you go to add chart element either from here or up here under chart design, add chart element, and you'll check the box for error bars. But then you want to go over here, scroll down and select more options. Sometimes it'll leave you in here where it's like, oh, what do you want the lines to look like? That's not what we really care about right now. So come over to the far option to actually alter the error bars. We then want to click custom, specify value, and you'll get this pop-up window. We want to delete what's present and then select the standard error values that we calculated. So click and drag to select all three of them. And then do the same thing for the negative error value. So click that, highlight it, delete, select the error values that we calculated, and now click OK.
Got it. Thanks.
Yep, you're welcome. We also want to add axis titles, so we're going to check that box also. And we have O2 consumption and cell type. And this is a totally acceptable figure for this data. For data display 3, now we have count data. We have whole discrete counts, and we've got cell type versus membrane lipid type, right? So we were saying before that eukaryotes use cholesterol, but prokaryotes use haploids. And then we kind of expect the isolated mitochondria to mimic prokaryotes, to be a little bit more similar to them because they have a more similar evolutionary history. So we want to select the data, all of it. We don't have to calculate anything, but don't include these. Those are kind of just for us. Those will be the labels for the axes in a minute. So select all of the data, insert bar. There's a couple different bar charts that you could make with this, honestly. And it's, you know, data displays are kind of like graphic design in a way where it's really, what's the message you're trying to say? And what do you think is the most effective way to say that? So I could select this, but I don't think it's the best figure. I don't think it's the best method for showing these data. So I'm gonna delete that and insert a different one. I think this might be a useful one, the stacked bar or stacked column, or this 100% stacked column, which gives us proportions. And I actually prefer that, so I'm gonna select it. But looking at this, I still don't think this is quite right? We have the membrane lipid type on the x-axis and the cell type as the colors. Again, this kind of comes back to what are you trying to emphasize? Here, I think we're actually trying to emphasize the cell type and the kind of membrane lipids that may be present. So I'm actually going to come up here to this switch row column and click that. And now I have isolated mitochondria. I have cell type on the x-axis, and the colors are the membrane lipid type. Yes, in this example, the explanatory values are categorical. So just like what we did with the other one, the other two, we're gonna go ahead and change the title and add some axis labels. So we can say the proportion of membrane lipid in 3 different cell types. I'm gonna add axis labels. Say proportion, or I guess technically it'd be more accurate to say percentage, and then cell type. And there you go. That's a pretty good figure to display this data, or these data, I should say.